Welcome to our educational video on enzymology. By the end of this video, you will be able to learn about different types of enzyme inhibitions with its mechanisms and examples. Enzyme inhibitors are substances that reduce the rate of an enzyme-catalyzed reaction either by blocking the active site or altering the enzyme structure. The inhibitor may be organic or inorganic in nature. And there are three main categories of enzyme inhibition: reversible, irreversible, and allosteric inhibition. In reversible inhibition, the inhibitor molecule binds to an enzyme and temporarily prevents it from catalyzing a reaction, but the effect can be reversed. And there are three main types of reversible inhibition: competitive, non-competitive, and uncompetitive inhibition. Competitive inhibition. Here there's enzyme, substrate, and inhibitor. The inhibitor resembles the substrate and competes for the active site of the enzyme. The inhibitor, which closely resembles the real substrate, is considered substrate analog. The inhibitor competes with the substrate for binding at the active site of the enzyme but does not catalyze the reaction. The relative concentration of the substrate and inhibitor, along with their respective affinity for the enzyme, determines the extent of competitive inhibition. As long as the competitive inhibitor occupies the active site, the enzyme is unavailable for the substrate to bind. Increasing substrate concentration can overcome this inhibition by outcompeting the inhibitor. In competitive inhibition, the Michaelis constant value increases while Vmax remains unchanged. For example, succinate dehydrogenase enzyme is inhibited competitively by compounds similar to succinic acid like malonic acid, glutaric acid, and oxalic acid. Uncompetitive inhibition. Here there's an enzyme, substrate, an inhibitor. The inhibitor has no structural resemblance with the substrate, and it does not interfere with the enzyme-substrate binding. The inhibitor binds at a site other than the active site on the enzyme surface. Binding of the inhibitor impairs the enzyme function. The inhibitor generally binds with the enzyme as well as the enzyme-substrate complex. Increasing the substrate concentration will eliminate competitive inhibition, but it will not eliminate non-competitive inhibition. The Vmax is reduced, but the Michaelis constant value is not changed. A competitive inhibition. Here the inhibitor binds to the enzyme-substrate complex, but not the free enzyme. Here both Vmax and Michaelis constant value is decreased. It is seen in phenylalanine inhibition of placental alkaline phosphatase. Irreversible inhibition. In this type of inhibition, the inhibitors covalently bind to enzymes irreversibly, inactivating them. These are usually toxic substances that poison enzymes. For example, disulfiram is used to treat alcoholism, which inhibits aldehyde dehydrogenase, leading to acetaldehyde accumulation and aversion to alcohol. Similarly, other inhibitors like iodoacetate, diisopropyl fluorophosphate, and organophosphorus insecticides irreversibly inhibit enzymes, affecting their functions and potentially causing harm. Suicide inhibition. Suicide inhibition is a specialized form of irreversible inhibition. This inhibition is also known as mechanism-based inactivation. The inhibitor makes use of the enzyme's own reaction mechanism to inactivate it. Here there is enzyme in a reversible inhibitor. This inhibitor binds to the enzyme, and then by the enzyme's reaction it is converted to more potent, irreversible inhibitor. So the suicide inhibition involves an enzyme converting a reversible inhibitor into a more potent, irreversible form binding permanently to the enzyme. Allosteric inhibition. Here there is enzyme, substrate, and an inhibitor. The inhibitor molecule binds to an enzyme's allosteric site, causing conformational change that reduces enzyme activity. By altering the enzyme's active site shape, the substrate cannot bind to the enzyme. Unlike competitive inhibition, allosteric inhibition is non-competitive, meaning the inhibitor doesn't resemble the substrate, affects the enzyme's function indirectly. Allosteric inhibition is crucial for feedback regulation in metabolic pathways, allowing cells to adjust enzyme activity based on the concentration of specific molecules. Feedback inhibition. It is a control mechanism in biological systems where the end product of a metabolic pathway inhibits an enzyme earlier in that pathway, preventing overproduction. For example, here is a reaction in which substrate is converted to product B by enzyme 1, and the product B is converted to product C by enzyme 2. The product C is the end product for this reaction. So when there is overproduction of product C, then it will inhibit enzyme 2 and can also inhibit enzyme 1, preventing overproduction. Typically, this happens via allosteric inhibition, where the end product binds to an allosteric site on the enzyme, altering its shape and reducing its activity. Feedback inhibition, unlike competitive inhibition, acts at a regulatory site, not the active site, making it efficient and reversible. This allows cells to quickly adapt to changing needs. You can get access to the YouTube videos along with notes and MCQs through our website link given in the description. If you found this video helpful, please like, share, and subscribe.